Hi, everyone. Thanks for tuning in and welcome to the Masterclass series. My name is Akelia Wap-Adumbo, and I am the Associate Director for the Technical Applications team in the Americas here at Oxford Nanopore. Today, I have the pleasure of kicking off this Masterclass series. Throughout the series, my colleagues will take you through all aspects of our platform and best practices for running an experiment from sample to answer. Our teams have curated specific topics within the Masterclass series that will guide you through our streamlined workflow that is comprised of 4 steps: preparation, sample and library, sequencing, and analysis. First, preparation, preparing the sample and then the library. The lessons include this Masterclass, guidance on how to extract high-quality DNA and RNA, and selecting the right library prep workflow for your experiment. Second, sequencing. That masterclass provides step-by-step guidance to loading a PromethION flow cell that will highlight transferable best practices to keep in mind for loading any of our flow cells. Third, the data analysis session discusses how to analyze your Oxford Nanopore data. And as a bonus, we have additional masterclasses that discusses how to sequence full-length plasmids in your own lab and how to call variants and methylation across the human genome. Let's get started. Here are a few key learning objectives that will be covered in this masterclass. We'll begin with an introduction to how Oxford Nanopore sequencing works, followed by the benefits of this technology. We'll go through how to plan your experiment with sample to answer in mind and some key considerations. Lastly, I'll share some online resources to provide additional support. Let's start with how Oxford Nanopore sequencing really works. As you might already know, at the heart of the technology lies the nanopore. A nanopore is essentially a very small hole that functions as a gateway between 2 systems, facilitating the transport of molecules from one side of a membrane to the other. In our system, we use protein nanopores that inherently have different shapes and properties that can also be enhanced through clever mutagenesis. We have selected the pores with the best features for sequencing on our platform. So let's dive into the details of the system. Here in blue, you can see a nanopore embedded in a supporting membrane. When an electrical potential is applied to the system, ions can flow through the nanopore. The current produced by this ion flow can then be measured. In purple, that's the motor enzyme, which is bound to a DNA molecule. The motor enzyme is added during the library preparation stage, and as you can see, it unzips the double-stranded DNA molecule and controls the movement of one strand as it passes through a nanopore. As the analyte, DNA or RNA, passes through the nanopore, it causes a distinct disruption in the flow of ions that is measured on our devices, captured as an electrical signal, and then interpreted by our base caller to determine the sequence of the DNA or RNA strand. As soon as a strand has passed through a nanopore, that nanopore becomes free to start sequencing another strand. Now that you've seen how Oxford Nanopore sequencing works, what are some of the benefits? Well, unlike other platforms that rely on synthesis, our approach reads molecules directly by monitoring disruptions in ionic current as they pass through a nanopore, allowing us to avoid many common limitations of other platforms. As a result, we can directly sequence DNA or RNA without the need for PCR, so there is no PCR bias. Moreover, it is possible to detect base modifications directly. Oxford Nanopore sequencing is read length agnostic, delivering reads from short to the ultra-long. Additionally, real-time sequencing enables real-time base calling and analysis without having to wait until the end of a sequencing run for answers. Finally, the chemistry underpinning the technology is highly scalable. The uniqueness of our platform, coupled with streamlined and robust workflows, facilitates diversity across investigations and techniques to address any of your sequencing projects. With Oxford Nanopore sequencing, you can access comprehensive genomic insights versus a limited view, which is common to sequencing by synthesis platforms. As we are not a sequencing by synthesis platform, access to longer, unbiased, information-rich data that is vital for assembly identification, variant calling, SNPs, SVs, and phasing, just to name a few investigations, permits inquiry into a broader range of questions, and more importantly, uncovering solutions and insights that once seemed elusive. Now let's switch gears and talk about how to plan your Oxford Nanopore sequencing experiment. At Oxford Nanopore, our sequencing workflow can be broken down into 4 consistent steps, and we have flexible options to support each step. We start with extraction of high-quality DNA and RNA. The samples are then prepared for sequencing using an Oxford Nanopore sequencing kit during library preparation. Next is real-time sequencing, and last is data analysis. Each step is explored in greater detail in the dedicated masterclasses that follow in this series. While this masterclass focuses on consideration at each stage of the Oxford Nanopore sequencing workflow, we also offer end-to-end protocols or application workflows. These application workflows provides tailored step-by-step guidance from sample to answer to help you achieve your specific experimental goals. So if you're looking for a complete start-to-finish solution, these provide everything you need. You can access our application workflows within the documentation section of our website. Currently, our workflows include human variation sequencing from a range of sample types, single-cell transcriptomics, nanopore-only telomere-to-telomere assembly, nanopore-only microbial isolate sequencing, rapid metagenomic sequencing for pathogen surveillance, and whole plasmid sequencing. When planning an experiment, we often start with our sample and how it should be extracted. We then envision a straightforward path where the extracted DNA is turned into a sequencing library, sequencing happens, the data is analyzed, and the scientific question is answered. In reality, things are a bit more complex. Let's say it's like a two-way conversation, and we recommend starting from the end and working your way back. In most instances, the bioinformatics requirements of the experiment will dictate the data that is required, for example, throughput versus depth of coverage, which in turn dictates the way in which you prepare the library. The requirements of the library prep could then dictate the type of extraction that you do. With that in mind, let's take a look at some additional considerations when planning your experiment. First of all, what are the experimental goals? This could be, for example, assembling a whole genome or species identification. Perhaps you wish to characterize variants or even phase your data. Next, what are the sequences of interest? Is this a single genome or a metagenomic sample? Will we be sequencing the whole genome, or are we interested in a specific region of interest that might benefit from a targeted approach? The size of the targets or genome is also an important consideration. Depending on the amount of data you need, you might wish to sequence a single sample or many in a multiplex fashion. So then what does the sample look like? We have solutions for DNA, native RNA, and cDNA, as well as options for different input amounts and varied fragment lengths. And last but not least, what other practical considerations are there? Is a shorter turnaround time required? How much data is really needed? Now that we have all those thoughts swirling around in our minds, let's look at the options we have at each step of the workflow so you can choose what best fits your experimental goals. Step 1. Preparing the input sample. This involves making a decision on what extraction method is best suited for your needs, again, with the data analysis in mind. Fragmentation and size selection can be applied to optimize for outputs and read lengths you need. We have a growing repository of validated protocols to provide some benchmarking and to set some expectations for later steps in your experimental workflow. It is essential to properly qualify and quantify your starting input. Step 2. Preparing the library. Here's a quick look at our family of sequencing kits, and as you can see, there's something for everyone. We recommend refining the kit options based on a few parameters, such as those shown here on the left, to help you make the best choice. There are kits for targeted sequencing and barcoding kits for multiplexing. There are PCR-free options, thus enabling the preservation of epigenetic signals, and for low input amounts, there's the option to use PCR. Real-time nanopore sequencing also enables adaptive sampling, which is a unique feature to our platform, and allows you to enrich or deplete regions of interest within the sequencing run itself, so at the sequencing read level. Additionally, you can unlock methylation sites without the need for any special library preparation modifications or steps. Step 3. Sequencing. We have a robust fleet of sequencing devices that cater to the scope, scale, and throughput for your required projects. The device you choose depends on the data output your experiment requires and the number of runs you need to perform. The MinION and GridION devices run MinION flow cells, whereas the PromethION devices run the higher throughput PromethION flow cells. The MinION device can perform a single sequencing run at a time, whereas the GridION can run up to 5 independently or simultaneously running flow cells. The PromethION family of devices has options for 2 or 24 also individually addressable positions. Our goal at Oxford Nanopore is to enable the analysis of anything by anyone, anywhere. Not only are our devices scalable, but we have also demonstrated their portability across diverse and even extreme locations around the globe, from deep-sea oceans to rainforests and even in space. Researchers are using Oxford Nanopore sequencing to sequence in low-resource settings with limited access to lab equipment, as our devices can be set up wherever and whenever they're needed. This has been critical in outbreak surveillance, where our devices have been used in real-world responses to outbreaks including influenza, Ebola, Zika, and SARS-CoV-2. This enables public health organizations to continuously monitor other viruses in efforts to launch more expedient solutions. And finally, step 4, data analysis. Similar to the other steps in our streamlined workflow, we provide a range of data analysis options, and finding the right one is easy. The Minnow software executes, monitors, and base calls your data. For further analysis, the Epitome platform provides easy-to-use, pre-configured workflows for a wide range of applications. These point-and-click workflows can be run locally or in the cloud via the intuitive user interface, without the need for prior bioinformatics experience. If you have more advanced experience, you can choose to run the workflows via the command line or build your own pipeline from our tools available on GitHub or developed within the Nanopore community. EliseION offers an all-in-one device with fully automated solutions ideal for a broad range of genomic applications with no previous genomics expertise necessary. The streamlined automated workflows minimizes hands-on time while also reducing turnaround times. Getting set up for sequencing is simple. Within each of our protocols, we provide an experiment checklist to be sure everything is in place before you start. Once you have all the items in place, you can start with the software installation and hardware check. Then move along to the flow cell check before really getting into the fun of things and preparing and sequencing your own libraries. If you're new or returning to Oxford Nanopore sequencing, it is highly recommended that you complete a control run. This will establish a baseline of performance within your hands, solidifying key nanopore sequencing steps, and will give you the opportunity to get familiar with our protocols and our analysis options. This all provides a better understanding of your existing computational needs and ensures best success for your actual experimental runs. Now that we have all of the pieces of the puzzle together, let's look at an example of an end-to-end solution, the Pathogen metagenomics workflow. Metagenomic sequencing is a key technique used in the identification of potential pathogens, providing important insights for outbreak surveillance that could inform public health measures. However, metagenomic sequencing using legacy short-read technologies can limit confident identification of microbes, as the short-read output makes it difficult to distinguish between closely related strains. Moreover, the need to batch samples results in a lengthy turnaround time. Oxford Nanopore offers a rapid end-to-end workflow for the metagenomic sequencing of microbial pathogens. Supporting reads of variable lengths, microbial genomes can be thoroughly characterized, delivering fast, accurate identification of bacterial, fungal, and viral pathogens from a single sample. The approach enables rapid access to data, which is critical for outbreak control. Here's an overview of the end-to-end pathogen metagenomics workflow, starting from respiratory samples. The protocol begins with extraction using a split approach to separately extract viral and acellular nucleic acids and bacterial and fungal DNA. The viral workflow incorporates a reverse transcription step following RNA extraction. Next is library preparation using the rapid PCR barcoding kit. This provides a fast method of preparing multiple samples for sequencing on a single flow cell. For sequencing of up to 22 barcoded samples in multiplex plus 2 controls, we recommend a MinION flow cell, which is compatible with either our MinION or GridION devices. Finally, for data analysis, the metagenomics workflow in Epidemy produces accurate identification and abundance information for the microbes present in the sample. Now let's take a look at the steps involved in a bulk transcriptomics experiment as a worked example of the Oxford Nanopore sequencing workflow. Uncovering RNA diversity is key to understanding biological mechanisms. Alternate transcript usage, for example, is a source of variation between normal and diseased tissues. However, short-read sequencing techniques often cannot span RNA transcripts in full. This can lead to missed novel transcripts and impacts the precise identification and quantification of isoforms. In contrast, the streamlined Oxford Nanopore cDNA sequencing workflow generates high outputs of reads that span full-length transcripts, allowing you to detect and quantify complete isoforms and fusion transcripts, and also measure poly-A tails. This information enables differential isoform expression analysis, providing vital information for mechanisms of disease research that could be missed by legacy methods. Here's the end-to-end workflow for bulk transcriptome analysis using cDNA sequencing. In this workflow, starting from a blood research sample, the QIAGEN PaxGene blood RNA kit is used to extract high-quality RNA. This is then prepared for sequencing using the cDNA PCR sequencing kit, which is optimized for high outputs of full-length transcripts. For sequencing, a PromethION flow cell is ideal, delivering over 100 million reads per flow cell for isoform level analysis. Lastly, the Epidemy transcriptomics workflow enables isoform and gene fusion identification, assembly, annotation, and differential gene analysis. Details on a direct RNA sequencing workflow is reviewed in the Library Prep Masterclass. Before we end this session, let's take a look at some online resources. Everything you need is available on our website, from resources on the Getting Started page to our extraction and library preparation catalog within the documentation section, and information on data analysis. Together, these will shape and refine all aspects of your workflow. And with that, we've come to the end of this masterclass. To summarize today, we have learned how nanopore sequencing works and the sample-to-answer solutions available, both manual and automated. We looked at what to consider before starting your experiment and how best to prepare your samples, always taking care to customize and/or optimize along the way. We next looked at a range of library preparation kits, sequencing devices, and data analysis options available to suit your experimental goals, and putting it all together with one of our end-to-end workflows. I hope that you have found the topics discussed in this masterclass beneficial as you continue on your Oxford Nanopore journey from sample to answer. Be sure to view the additional masterclasses within this series for further guidance and best practices. Until next time, thanks for tuning in.